We used to just use some traits. That's something that you should have seen in animal diversity, right? Based on the common traits, we split the groups into this or that. But really, the organization depends on the pattern of their development. So eukaryotic organisms have a nucleus and a chromosome that undergoes mitosis. We will focus on this course for Vivo in the metazoan group, which is the group that we have here in the screen, which they go through gastrulation. If it does not go through gastrulation, it's not an animal. Okay. So in this course, we will be looking at specific examples of few protostomes because they possess a notochord. You're going to see me in the lab talking all the time about the notochord and the important role of the notochord in development. And then we will be looking at deuterostomes, okay? So in here, we're going to be looking at the sea urchin that we have right here. We're going to be looking at fish too. We're going to be looking at amphibians, birds, or mammals. Those are the 5 groups that we're going to compare just to see that they have a lot of commonality in the way how the different germ ledgers are created, and at the same time the patterning of the body is very similar, independently whether it is a sea urchin or whether it is a mammal. So the last is in this group over here. So excuse me, my throat today is a little scratchy. Cleavage in sea urchin is determined in part based on the content that it has, okay? So in sea urchins have what we call radial holoblastic cleavage. Remember, we already defined these terms. Radial because it goes in angles sometimes, and holoblastic because it goes from the animal pole to the vegetal pole. This the whole thing, holoblastic. Okay, meaning that it's an egg with little vitellus, right? Because that was the other trait that we defined. So it can go all the way through. And the cleavage go all the way through the entire egg. In here, we have the first and the second cleavage. These cleavage are made in right angles to one another. So the first one was this one, what we have here, and the second one is perpendicular to the first one. Okay, so right angles. So cleavage fiber pass through the animal and the vegetal pole, and we call this one meridional cleavage. Okay, because it goes from one end to the other end. The third cleavage, that is the one that we have here in this section, is equatorial. Okay, and it really separates the animal portion pole from the vegetal hemisphere pole. Okay, so one meridional, second meridional, third equatorial, segmenting the animal from the vegetal hemisphere. The fourth cleavage, the one in here, the 4 cells of the animal hemisphere divide into 8 blastomeres. Okay, so we have 1, 2, 3, 4, the 4 that are from the third cleavage, and they divide into double, so 8 at the end. Okay, and each have about the same volume. These cells are called mesomeres. The 4 cells that are on the vegetal hemisphere undergo an equatorial cleavage, but this cleavage is unequal. Okay, so this is unequal equatorial division from the vegetal half, because we're going to have the ones just underneath the mesomeres are bigger, and at the very end of that hemisphere, are smaller. So these one are mesomeres, these are macromeres, and these are micromeres. Okay, so still this is 8, but these ones are 4 and 4. Okay. Now, this is a 16 cell stage, right? So 8 mesomeres will divide then equatorially. This is, you see the fate maps in these ones with the color change, the pale and the darker blue. So that's the equatorial line where you're gonna divide the 16 cells on top, the 8 cells on top. So now we're gonna have 16. And this one, we're gonna have a tier that is the animal 1 and a second tier that is the animal 2. Okay? Stagger above one another. The micromeres, so the little one is in red, also divide, producing a small cluster beneath the larger tier. So these ones are gonna divide non-equatorially as the mesomeres did, but they're gonna divide meridionally. So we're gonna have, here we have 4, now we're gonna have here 8 and 8, okay? Now, the 6th cleavage, the animal pole divides meridionally. We'll see here. So we have a double of each ones. And the vegetal pole divide equatorially. So here we have a more equatorial subdivisions. The 7th cleavage now, the pattern reverses. And at that state, at the 7th cleavage, we have a blastula. So from cleavage number 7, the pattern of cell division becomes much more less regular, as we have seen in here. Meridional, meridional, equatorial. Meridional, equatorial, equatorial, meridional. And after that, it's just absolutely a mess. So we don't count anymore the pattern of segmentation from that section. So 7th stage, I say we have a blastula, right? So the blastula cells normally, as division start going, so here is 1, this is the micropili right here, this is the fertilization envelope, we have the zygote 2 cells, then we have 4, then we have 8, so we have here the micromeres here underneath, and you see as it divides, we start having a little cavity in the middle, right? The space that is void. And that is the blastocele. So it's a blastula. Blastulas have a blastocele. It's the cavity that is inside. Okay. So now that the cells are all more or less about the same size, because as it goes, it starts changes. Cell division of the micromeres, not the macromeres, but the micromeres start slowing down. Okay. Every cell contacts the proto-initius fluid. So it's the fluid that is inside over here of the blastocele. Okay. And there is the hyaline layer on the outside. Okay. The cell's gonna continue dividing. We see here they are still big, and here we start getting smaller, and in here continue to divide. Remember, they don't increase inside the cells, but we're gonna get less and less, more cells, less cytoplasm. Okay. So this rapid and invariant cell cleavage that it goes through the 9th and the 10th cell division can vary depending on the species. Cell-developed cilia. Just wait a second. I just want to allow them to come. So we're going to start seeing some cilia that is going to appear at the surface of this blastula, okay? And that is what we call the ciliated blastula, and start rotating inside of the fertilization envelope. Let's start rotating inside. The cells that are located at the vegetal pole, and we know that is the vegetal pole because we have the micromeres, right? So the cells that are found at the vegetal pole start to differentiate. And they start, and here we see again the micromeres, and here we see this is the position where those micromeres were. And what we see here is that those cells start to thicken, and that what becomes the vegetal plate, right there where the micromeres were, okay? So these cells that are found in the animal pole start synthesizing and secreting a hatching enzyme that will break through the fertilization envelope, and the embryo is now a free swimming hatch blastula because it has the seals, okay? So if we look at the fate map of the different cell lineages as the egg continues to divide, and this is about 60 cells, 60 cell stage. Most of the embryonic cell fates are now specified, okay? But the cells are not irreversible committed, meaning that if you transplant them to another part of the embryo, they can give rise to other cell types because they are still pluripotent, okay? So remember, it's not totipotent. Totipotent can become any type of cell, right? But pluripotent has a wide variety, but it's still limited. Okay, so the animal half, so all of this section in blue, okay, of the embryo will give origin to ectoderm. Remember, blue color from now on means ectoderm, okay? So the animal half will become the ectoderm. So that's the animal 1 and animal 2 part, okay? VEG1, which we have in here, layer, produces cells that can produce ectoderm and endoderm. That's why it has the blue color and the yellow color. Remember, yellow is always endoderm, okay? VEG2, the second tier over here, give rise to cells that can populate different structures. We have endoderm in yellow, okay? Which also gonna have the body wall, meaning the coelom, the internal mesodermal wall, body wall of the individual. And we're going to have also the non-skeletonic mesenchyme, which is seen here in the red color. So it's also known as the secondary mesenchyme, which will generate muscles and pigment cells and some immunocytes too. Okay, so that's the second tier, the VEG2. The second tier of the micromeres, which are the larger ones here, the ones in red, okay, produce the primary mesenchyme of the skeletogenic mesenchyme. And those are the ones that are going to be forming the larval skeleton. The second tier of micromeres, which are the small micromeres, the ones that are right here, contributes to the coelom, the cells to the coelom, from which tissues of the idol and during metamorphosis also, these are the ones that are going to help. Okay. Now, the signal, because all of this is about signal. Okay. the molecule responsible for specifying the micromeres here and their ability to induce neighboring cells is our friend beta-catenin. Okay? Just to remind, beta-catenin is a transcription factor activated by the Windlass pathway, the WNT pathway. Okay? So the accumulation of this transcription factor specifies the vegetal health of the embryo, and that's why we're going to start having the vegetal plate, because these are the micromeres that's going to start differentiating. Okay, so beta-catenin gives rise to micromeres, their inductive ability by activating the genes that produce the inductive signal. So we saw beta-catenin in the planaria paper, we saw beta-catenin in the regeneration paper of the fish, and here again, beta-catenin. So it's one of the first signals that the embryos are receiving for determining the fate of most of their cells. Is this figure well explained? Is it clear? Okay. Okay, so the fate of different cell layers are determined in two-step process, as we saw previously. So the large micromere, these are autonomously specified, and we already talked about the autonomous specification, right? So the information comes from the vegetal pole and becomes incorporated in the blastomeres. So the cells become escalogenetic mesenchyme and become the larval skeleton, as we say prior. So here we have the micromeres in red. Here, this is the free swimming blastula. We see the cells, right? And we see how they go inside, right? And we want to see all those movements in a couple of minutes. And then become the skeleton. It is pretty, right? The second step, the autonomously specified large micromeres are now able to produce paracrine and juxtacrine factors, which means that if we have paracrine and juxtacrine factors, what type of a specification we are talking about? Juxtacrine is to be side to side, right? To induce. And paracrine is one cell produces something that is necessary for that same cell or for a cell that is close by, right? So it's conditional specification, okay? So that's the second step. So the first, the large micromeres are autonomously specified. It has everything they need to become the escalogenetic mesenchyme. But the second step is that those micromeres will now, that have been differentiated, produce paracrine and juxtacrine factors through conditional specification. So they are going to decide the fate of the cells that are surrounding them. Is that concept clear? Okay, so the micromeres produce a signal that tells the cells above them to become endoderm and induce them to invaginate into the embryo. So this is how we have here, this fates in here, this cell's going to start invaginating. Remember that movement, going inside is like poking your finger into a balloon, okay? And this is how it's going to go inside, and this cell's going to reach the other end of that blastula, okay? So the ability of micromeres to produce signals and change the fate of cells is very important in development. If they are removed, this is what we have in B here. So we only have the animal hemisphere, okay? Then we don't have it. We just have the induction of the external ectoderm, right? And the ectoderm was supposed to produce cells, and this is what it does. But we don't have the escalogenetic part into, because that is induced by the micromeres, okay? In C, we see that if the micromeres are placed with isolated animal hemisphere, we are missing the VEC1 and part of the VEC2. We're just putting the micromeres, right? So we know, we see in here that we're just missing that section, but we still have the induction of beta-catenin that is contained in these micromeres, and we are able to produce a plateaus, okay? But the endoderm is missing. It's not there because we are missing that signal, the competent cells. Remember, this type of induction depends on whether the cells are competent to accept the signal and change their fate, okay? So a little bit more about the signal in this sea urchin. So the initial regulation input are two transcription factors. The one that I mentioned prior, beta-catenin, but there is also a second one that is dishevel. We talked about dishevel already. We explained the transcription factor for dishevel, okay? So which two factors, beta-catenin and dishevel, are found in the egg cytoplasma and are inherited by the micromeres. So we see here the micromeres. That's the dark color is telling us this beta-catenin is specifically concentrated in those micromeres, okay? And this is from the fourth cleavage. These cells already have that fate. They are the ones to produce the signal. So it's very, very early in development. Fourth cleavage is just like 3 seconds after the first one. So it's very, very quick, okay? Dishevel is also found at the ventral cortex of the egg and normally prevents the degradation of beta-catenin. So that's the one that is surrounded, okay? And it's impeding that beta-catenin will be degraded. And normally, what happens is that the VEC2, the tier, normally going to have something that will degrade beta-catenin. So by surrounding this section over here, we want to prevent that degradation of beta-catenin in there. So beta-catenin specifies the micromeres. Beta-catenin accumulates in the nucleus of those cells fated to become endoderm and mesoderm, okay? So this is what we see. So all these cells that are supposed to have endoderm and mesoderm will express that beta-catenin, okay? So here we see that expression very, very, very clearly. If we treat embryos with lithium chloride, for instance, that allows beta-catenin to accumulate in every cell of the embryo and is capable of transforming the presumptive ectoderm into endoderm. And this is what we see here in figure C. So not only we have these micromeres that had high amounts of beta-catenin, but we see that it's everywhere, right? And then we have this larvae that is produced, but doesn't have the shape of the plateaus, right? It's just like a whole ball of endoderm, okay? But if experimentally beta-catenin is prevented to enter the ventral nuclei, this is what we have in here. There is just a little bit of expression, but not even. Okay, so these are the small micromeres. We don't see the accumulation of beta-catenin, and the vegetal cells fate are not specified, and the entire embryo develops as a ciliated ectodermal ball, and this is what we see. It's just a ball with cells. The cells, you see the cells here, they're very, very, very faint on that edge. Okay, so no formation of endoderm or mesoderm. Okay, so there are other factors providing input for the regulation of the micromeres, but I will not discuss them right now. Okay. Okay, so that was all about the blastula. the rapid division of cells to produce the blastocele. It's important to have the blastocele because the blastocele will separate ends, right? The animal versus the vegetable pole. And now we have the differentiation of that vegetable hemisphere induced by the beta-catenin, right? Of the disheveled. So that was the first path of induction. The second step is gastrulation. So the late sea urchin blastula, the one that is free swimming, okay, is a single layer of about 1,000 cells in the form of a hollow ball. So this is what we have in here. Here we have the cilia all around. This is the animal hemisphere, and this is the vegetal hemisphere. How we know that this is the vegetal hemisphere? Look at the thickening of that pole. It's thicker than this end, okay? And it start a little flat, it's not as round. So that's only 9 hours after fertilization, okay? So the blastomeres are from different regions of the zygote and have different sizes and properties. And we see that here, right? We have some that are a little bigger or smaller, depending on the position that they have. So the cells that become the endoderm, meaning the gut of the individual, and the mesoderm, meaning the skeleton of the individual, are still on the outside, right? This is the outside, and they need to go inside the embryo. And that's what happens during gastrulation. So in here, we're going to have a different movements, and we're going to start with ingression, okay? So shortly after the ciliated blastula hatches from fertilization envelope, the vegetable pole starts thickening, as we see in here, and flattening. The only way how you can thicken is by ingression. Remember ingression, you are duplicating the number of cells. They detach and go inside. Remember that movement, okay? If you don't, you better go back to those different movements because from now on, we want to mention them again and again. Okay, so I want you to be sure that you know what I mean when I say ingression. So at the center of the flat plate, this is what we see here at 9.5 hours, okay? A cluster of small cells begins to change, and we see it. We see here, they start to change, right? And in here at 9.5, we see a little more change in that one. So these cells will start extending and contract along a thin process that we call the filipodia, okay? So we don't see it too much in this image, but they are in there. So they're gonna start stretching, like pulling themselves up through filipodia. It's like a lasso. They just hold on something and then pull up, okay? So the cells then separate from the epithelial monolayer and ingress into the blastocele. So that's what they're doing in here, separating and ingressing, going inside, losing their connection to this layer over here in order to go inside, okay? These cells now that are in here is what we call the primary mesenchyme cells, and are all derived from the micromeres that were at the vegetable pole, okay? So these also can be called the eskelotogenic mesenchyme because they form the larval skeleton. So these cells become localized within the ventrolateral region of the blastocele. So we see here they start continue forming that primary mesenchyme, okay? And then we're gonna, is in the ventral portion, okay? And we're gonna see the next movement. So how they get there? Well, they need to go through the epithelial-mesenchymal transition. So in order to get inside the embryo, the micromeres need to go to the EMT, Epithelial-Mesenchymal Transition. This transition is possible, as we saw a couple of chapters ago, right? Because the cells lose their affinity to the neighboring cells and the hyaline membrane and gain affinity with a group of proteins that line the blastocele. So this is what I say the blastoceles plays an important role, okay? So in here, we have the blastocele. This is the basal lamina. This is the hyaline layer, and these are the cilia, okay? And here is the blastocele, and this here in yellow is the extracellular matrix. Remember that we say that the extracellular matrix can give some signals to the cells and maintain the integrity. So here we have the rebel cell. This is a rebellious cell that says, hey, I'm out of here. I want better for myself, right? And start losing the connectivity with the hyaline layer until it gets attracted. So this extracellular matrix is giving them signals to do that. And then they completely disconnect from the neighboring cells, okay? So proteins in the lining of the blastocele induce some of those cells to get out of there. Here, they fuse into syncytial cables, that's what we call, which are formed from calcium carbonate. These are spicles of the larval skeleton, and we're going to see them in the next slide a little more clear, I think. So we're going to see a couple of slides that shows them. So the primary mesenchyme cells leave the vegetal region of the blastocele. So in the view, we can see that hollow blastula in a couple of minutes. That shows how we lose that connection here with this dotted line that is the basal lamina, and we can change the fate of that cell. So 5 events are required for the transition for the ingression of the cells, and that's the ones that we have in the screen. One is the apical basal polarity. So the vegetal cells of the blastula elongates to form a thickened vegetal plate. This is in here, and in here we see that the plate is more evident, okay? Second is an apical constriction of micromeres. So the cells alter their shape with a change in the apical end. So go away from the blastocele, and this is, if I show you here, this is, you see, this is the apical shape that is changed to start losing the connection. Third is the basement membrane remodeling. So the cell pass through the basement membrane, and with the proteases they secrete, they gain the inside of the embryo, okay? 4 is the de-adhesion. So we have the catenaries that maintain cells together. Remember, we talk about the catenaries, how they are like the glue for the cells, okay? So it starts degraded and they become loose. There's a transcription factor that is called SNAIL, is responsible for the degradation or downregulation of the catenaries. So they lose connection due to this SNAIL transcription factor that is expressed in the plate, in the vegetal plate, and then cells are losing the connection. And then is cell motility. We have a transcription factor that is called the GRN, is gene regulatory network. That's the GRN stands for, that activate proteins that promote cell migration to the blastocele. So 5 steps. So these cells at the vegetal plate can lose their connection and gain the inside of the blastocele. And all of this just within an hour. It's amazing, no? Okay, so now that the cells have gained the blastocele, they need to start binding and migrating to form a structure that is very important, that is called the Archenteron. So to do this, they start clustering together. And this is what we have in image A. Okay, so we have, these are the cells that just migrated inside by ingression. Okay, and they start clustering. If I come back a little bit, you see them here, and here you see that they start clustering, right? So this image, we see the massive amount of beta-catenin in red, okay? And inside, we see the sclerogenic mesenchyme in green. These are the cells that have entered the blastocell by ingression. And we see that these cells accumulate where beta-catenin concentration is higher. Where is higher? At the plate, right? Even though we have a little bit on the edge, it's higher concentration of beta-catenin at the very plate, at the vegetal plate that they have. So they start accumulating a little higher. And we see here and here, right? So in B, what we have in here, we see that cells have fused together. They are all clustered, right? And they start elongated the filipodium. These little hair-like projections are the filipodium, which starts sensing the environment of the blastocell wall. So these proteins that are attracting them to migrate to the blastocell and determine the dorsal ventral side and the animal vegetal pattern of the ectoderm. So they know this gradient here and say, okay, I'm gonna bind here, but then I'm gonna pull another carbonate filipodium over here so I can pull from it, okay? So two signals are required for the migration of these cells in order to be able to come from here to go somewhere else. So we have the VEGF paracrine factor, so this one here, that is emitted from the ectoderm where the sclerogenic mesenchyme cells will congregate. And the second factor is the FGF factor, so the fibroblast growth factor, which is a paracrine factor, as we have described it prior, made in the equatorial belt. So where is that one? Over here, right? Between the endoderm and the ectoderm. So the sclerogenic mesenchyme cells will collect towards those points. So we see how we see these cells really accumulating when the fibroblast growth factor is found. So cells always are following a signal to tell them where to go. So I say that these cells will form a second structure. The first structure important was the blastocell, right? And then we have the primary mesenchyme. And now the second or third structure important is the Archenteron. So 3 stages are required for the formation of the Archenteron. First is the invagination of that vegetal plate. So we had our plate here. The cells have gained the inside. Now they are placed at that belt, equatorial belt. And now that section, as we see here, the transformation, they start invaginating, so poking inside, okay? The second step is the Archenteron increase in length. So cells start pulling and thinning. So still here we have the cells right on the side. Here is where invagination happened. And we're gonna have all those cables pulling these cells to the opposite end, okay? So the filipodia will pull this Archenteron towards the animal pole to make a continuous digestive tube, okay? So we're gonna go through the different steps. So the first stage of the Archenteron is invagination. So as the non-sclerogenic mesenchyme leave the vegetal region that it was found in this section, okay, of the blastocell, important changes are occurring to the cells that remain in that vegetal plate that was found in this region here, okay? So they start moving to fill the gaps caused by the ingression of the non-sclerogenetic mesenchyme. Next, the vegetal plate bends inwards and invaginates. The invagination takes time, so it's just a quarter and then up to the halfway into the blastocele, okay? So we want to see that in next slide, okay? Now, the invagination suddenly ceases. So a quarter or a half of it, and it stops, okay? And the invaginated region becomes now the Archenteron, which is the primitive gut. And the opening into the Archenteron is what we call the blastopore, okay? So that will become the blastopore. So let's see these figures, how it goes. So thickening, this is the equatorial section, and we see how cells are moving inside and moving inside, and this is about half of the blastocele cavity, right? And then it stops. And this is only 1 hour again. So very, very quickly still. What happened with that? Why it stops? So this is what we see in this fate map here. So the central portion is gaining its fate, right? So even though the micromeres are the ones that start moving these cells, and this is what has encircled, start moving inside by ingression, then these cells over here start also gaining the inside as invaginates, right? So it's now, it has a different type of information a little bit, because the small micromeres have already migrated by ingression, but these cells are remaining there, right? So they need to be told what to do. So the central point becomes non-sclerogenic mesenchyme cells. So these are what are a little bit more pinky color over here, okay? The concentric cells around becomes the foregut, midgut, and hindgut, okay? So these are the yellow thing with each of the color. Darker fore, medium yellow is midgut, and pale yellow is the hindgut, okay? And then we observe here that the veg 1 and veg 2, so these two sections over here, help for the formation of the different parts of that digestive system as they start invaginated inside the blastocele. The non-sclerogenic cells are the first one to invaginate. So we have these ones that invaginate and hence form the tip of the Archenteron, leading to the other cells into the blastocele. So these non-sclerogenic mesenchymes are the ones that are at the center of here, of our Archenteron. So they are the ones leading the growth of the Archenteron, okay? So these non-sclerogenic cells are also responsible for the pigmentation, the formation of muscles around the gut, and the formation of the coelomic pouches that the individual will have. The second stage of the Archenteron formation, after the invagination and after the brief pause that these cells are doing, is a continued invagination, but longer. So in here, the Archenteron increases its length by 2 to 3 times. So it was half of it, and now look at that, it started going. And you see here, just gonna see the spicules that is helping to attach to the wall of the blastocele to pull. And we see here how those cells continue pulling until they're gonna get to the roof of that blastocele. So the wide, short gut rudiment is transformed into a long, thin tube, right? We were thicker here, and then it becomes thinner over here. And this is accomplished by the cells of the Archenteron that rearranging and migrating over the flattening themselves. So they go and flatten. You see here, we see a little bit of roundness, and here it becomes more like a flat, very thin. Okay, so cell division continues producing more endodermal cells, which enter the embryo. And we see also the formation of the secondary mesenchyme, that is a second wave of non-sclerogenetic cells. And the Archenteron continue extending. So we see here, we only have a few, but here we see way more cells. Okay, so this is a second wave of mesenchyme cells going on. So this cell movement makes the cells to start intercalating between one another, and that's what we call the convergent extension, because they intercalate among themselves. So here we have early gastrulation with the initial invagination. We see here cells with different numbers. These are the ones that are leading the growth. And in here we start seeing just a little bit more of the intercalation of those cells as the walls start thinning to allow the elongation of the Archenteron. Is this clear here? Okay. The third stage in the formation of the Archenteron is the second wave of non-sclerogenetic cells from the tip of the Archenteron, and they remain there for a while. Okay, so these non-sclerogenetic cells cast out Phyllopodia, like we just are casting when we are fishing. Who likes fishing? I do. You like fishing? Yeah, Emma says yes. Annalise, you like fishing? So. Okay. Annika, you like fishing? Yeah, good. So they start casting the Phyllopodia. So here we see it, all the Phyllopodia going up, attaching to the roof of that gastrula, and attaches really tight to that wall of the blastocele. So they contract and pull the Archenteron toward the inner surface of the animal pulp. So these cells on the inner surface of the animal pulp appear to have a specific target proteins to which this Phyllopodia will attach. So there are specific proteins, okay? So at the top of the Archenteron, where the blastocele wall in the target region and the non-sclerogenetic cells disperse into this will become the mouth of the larvae, okay? And the initial blastopore becomes the anus of the individual. So the point where the Archenteron contacts the blastocele wall is the mouth, and the mouth fuse with the Archenteron to create the continuous digestive tube, and thus is the characteristic of deuterostomes. The blastopores form the anus. Okay, that's the trait of deuterostomes. So, in a summary, sea urchin cleavage is radial and holoblastic. Cell fate is determined by autonomous and conditional specification. Maternal beta-catenin is really important for the autonomous specification of the micromeres. Differential cell addition is important for gastrula. So that's how we can go through this epithelial-mesenchymal transition. Okay. And at the same time, they can adhere to the blastocele wall. Also, during gastrulation, cells from the ventral plate move into the blastocele by invagination to form the Archenteron. But don't forget, we also have the ingression of the cells, okay, at that point. And the non-sclerogenetic cells allow the elongation of the Archenteron by convergent extension. And that is how we obtain a sea urchin. In this case, I'm just going to show a little video about this sea biscuit life. Is about this, so it's very short video, but it's so cool. And at the very end, we have a sea urchin. Okay, now I need to come over here. Do you have any questions regarding the sea urchin development? Nope. Feeding on fish. Okay, so chapter 11 is about the development of amphibian first and then fish. So as I stated at the beginning of the course, early developmental stages is very similar in each of the vertebrate groups, and this is when we want to start looking at those similarities. Fish and amphibians are widely used for developmental studies due to the large amount of eggs that they produce. right? One clutch can have 500 eggs, right? And they have an external fertilization, which becomes very handy if we want to just induce fertilization ourselves to study all the different stages of development and the different things that can affect that development. And that also because the development of the embryo can be easily observed, because particularly in fish, most of them, the eggs are very clear, particularly in the Danio, the zebrafish, the eggs are completely translucent. It's unfortunate that we don't have the lab in person, because I normally rear them in the lab. So every time that you come, I put them the day before together. And in the morning of, I go to the lab and check how many eggs and how many are fertilized, which the stage we are. We take photos of it and things like that. So within a lab, you can see maybe the heart start beats, and then you see the blood formation and the pigments migration. Sometimes it just was fertilized an egg, and then you can see the cleavages as it go and the formation of the somites. So unfortunately, this year we won't be able to see it. We're gonna see a video on it later in the lab. So we're going to start this one with the amphibians dissection. So the entrance of the sperm during the process of fertilization causes a radical displacement of the egg cytoplasm. And that is a rearrangement of the cytoplasm of the egg. And so, as we said last week, this week. So fertilization this week, oh my God. Fertilization can initiate a lot of these rearrangements. So in here we see this is the point of entry of the sperm. As soon as we have the cortical reaction that we explained before, we see a migration of the cytoplasm, and like counterclockwise 30 degrees. And that migration here is really important to form this great crescent. And we're going to see how capital this great crescent is for the process of gastrulation, okay? So a single sperm can enter anywhere on the animal hemisphere of the egg. Only on the animal hemisphere, is not possible in the vegetal hemisphere. And this is the pigmented zone with melanocytes. So this is what we know, this is the animal pole, right? Because of the pigments that it has. So the point of entry of the sperm marks the ventral side of the embryo. So the belly of the embryo. Always the point of entrance of the sperm in amphibians mark that belly portion of the embryo, okay? The side 108 degrees opposite to the side of the sperm entry will mark the dorsal, meaning the spinal side of the embryo. So this is the back, and this is the dorsal, the ventral part of the embryo. In one cell embryo, the cortical cytoplasm rotates these 30 degrees that I mentioned here, okay, with respect to the internal cytoplasm. So this is what we see here in this C, okay? After the sperm entry, the cortical, meaning the outer cytoplasm, shifts that 30 degrees to the point of the entrance in here and forms that great crescent. Because the cortical cytoplasm of the animal pole is pigmented, this gray creates that light zone here. And that's what we call the gray crescent, just because the pigmented zone moves, and now this section doesn't have that pigment, okay? So that is the gray. And that is where gastrulation begins, okay? So the microtubular array organized by the sperm centriole at fertilization become important in initiated the dorsal ventral axis of the larva. So just by the entrance of the sperm, now we have dorsal ventral axis, period. Pretty easy to determine that way. Okay, cleavage. So like in sea urchin, cleavage is radial and holoblastic. Okay, as we saw in the sea urchin, it starts with meridional, right? First one and second one in right angles to one another. So still meridional. Now, because amphibian eggs are mesoleucithal, remember we defined them, that contains more yolk, right? Compared to the sea urchin egg, meso, medium, okay? We see that the second cleavage starts before the first one has reached the vegetal pole. Okay. So first division meridional bisects the great crescent and slowly progress from the animal to the vegetal pole. This movement is slow because of the high content of vitellousness that is mainly concentrated into the vegetal pole of the egg. The second division, as we saw, is in a right angle to the first and is also meridional. Because of the yolk, then the second begins before the first one is completed. Okay. The third cleavage that we see here is equatorial, but is displaced. Okay. The equator is here, normally where this great crescent is placed, right? So it's displaced in this case. And this is because of the yolk content. The animal section has less yolk than the vegetal pole. So this is why the third equatorial is displaced. It's not at the equator. And here we have a little bit more of vitellus in that section. So the embryo after the third division is composed of 4 similar group of cells, okay, here, that we call the micromeres at the animal pole. This is a rapid cleavage. And we have 4 larger cells, 1, 2, 3, and 4 on the other end, that is called macromeres in the vegetal pole. And continue. This is slow cleavage due to the presence of this vitellus, okay? As cleavage progresses, the animal region becomes packed with numerous small micromeres, while the vegetal region is composed of relatively small amount of yolk of macromeres. So just to recap, first division is meridional, that bisect the great crescent and slowly progress from the animal towards the vegetal pole. Second division is at the right angle to the first and is also meridional. Because of the yolk concentration of the vegetal pole, it slows the cleavage. The second division begins before the first one is completed. So here, that's what it happens in there. The third is equatorial displace. And this unequal holoblastic cleavage established 2 major embryonic divisions. So the rapid in the animal pole to form the micromeres, and the vegetal pole, the macromeres. Okay. There are numerous cell adhesion molecules that keep the cleavage blastomers together, because we see them here clump, right? And one of the most important of these molecules is the epicadherin. So the mRNA for this protein is supplied in the oocyte cytoplasm. Remember that the egg cytoplasm contains so many stuff that are required for the growth and division of the embryo. Okay, so we are just on the third cleavage here, and we can see right away a little bit of a fate map. Okay, so The animal hemisphere, excuse me, cells will be raised to the ectoderm. This is our ectoderm, always in blue. The vegetal hemisphere will give rise to the endoderm. Okay. Cells that are beneath the blastocele cavity will become the mesoderm. So all of this is a little bit of mesoderm in red. Okay. Now, cells opposite to the point of a sperm entrance will become the neural ectoderm. So as we saw prior, the sperm entered somewhere over here, right? So in the opposite side, that's the neural ectoderm. And we're also going to see at this point in a little while the notochord mesoderm and the pharyngeal head, okay, endoderm. So a lot of things going to change in this section. So the blastula formation in amphibians is when we have about 16 to 64 cells. Then it's become the morula. So very early in development changes, right? In the sea urchin, it took a little while to have the morula. We didn't really see a morula in sea urchin, but in amphibians, we have a morula, and it comes from the Latin from mulberry. It looks like a mulberry, exactly, okay? At the stage of 128 cells, the blastocele appears. This is our blastocele, and we have about 168 and 28 cells, okay? The first cleavage furrow widens in the animal hemisphere to create that small intercellular cavity. So we're gonna see some cuts that is just a tiny hole, and as it continue to divide, we have, and you see that blastocele is more placed towards the animal hemisphere and not towards the vegetal hemisphere due to the presence of the vitellus in this section, okay? So this cavity expands during the subsequent cleavages, and then we have this big blastocele. The blastocele accomplishes two main functions. The first one is that permits cell migration during gastrulation. That's what we're going to be seeing next. And the second one is that prevents cells beneath the blastocele from interacting prematurely with the cells above it. If the cells interact prematurely, their own tissue will be formed. Remember, is an induction pathway, right? So you need to be in the right position at the right time to be able to form the right type of tissues, okay? So cells from the roof of the animal pole, if transplanted to the floor of the blastocele, it will induce to form the mesoderm instead of the ectoderm, for instance. So these are type of things that happens if things goes wrong. So this is the overall fate map in Synopus. So in the amphibian blastula, it must bring inside the embryo the cells are destined to form endodermal organs. But we know they are here at the bottom. So they need to gain that inside of that embryo. Then they need to surround the embryo with the cells that are capable of forming ectoderm, because right now the ectoderm is only here in the animal hemisphere, okay? And then it needs to place the mesoderm in between both of them as a sandwich, okay? So cells have different fates depending whether they are located in the deep or in the surface layers of the embryo. And here we have all of these ones are simply on the outside. So we need to do all those movements. For instance, the mesodermal cells are deep in the embryo, while the endodermal and ectodermal are at the surface. So endoderm needs to go in and then place in the right position with this ectoderm. So most of the precursors from the notochord and the other mesodermal organs are located beneath the surface of the equatorial region. So this is the ones here, the lateral plate mesoderm. This is the somite and notochord. These are the somites, and these are the heart. So we need to place all these sections in the right position. And so this is when gastrulation is very important to achieve getting the superficial endoderm precursors into the interior and the deep mesodermal precursors outward to a location in between the ectoderm and the endoderm, okay? And we are going to stop today right there. Where am I? Here, because I want to give the complete section on, now I need to escape from here, of the gastrulation in one shot instead of dividing them. It's better just to have some fluidity on this lecture. So there is a question in the chat. I didn't even pay. No, right. My hair just wrote no, but I do not know to what. Okay.
No, if you have any questions, you asked that before.
Oh, okay. You were too quick. My screen takes it's a little lag between I just say go into full mode and then you appear again into this screen instead of the other screen. Because I have you all in my other screen, and in front I only have my the PowerPoint. So tomorrow we see each other at 1 for the lab. I will release the notes for the lab. I think it's only with Maher that I have been having this conversation about the manual. Who doesn't have the manual yet for the lab other than Maher?
I don't. I'm still waiting for it.
Okay. So as I say, I cannot make this available because I will be in violation of the copyrights, and that's something very bad because the university will be, I will be charged of guilt. So I found a little way how I can try to provide you with the lab notes for the moment. Okay, we'll see how it goes. So I needed to convince IT to help me. It's good that I have good friends in IT that give me the tricks. So the way how it's going to work is that you're going to have access to them, but you cannot extract, you cannot print, you cannot copy, paste, nothing. Just see it on the screen. Okay? And that's the only way how I can do it. Because in that way, I am not giving access to a copy that you can keep and share, right? So I had that discussion with the editor. I say, stop, don't tell me anything. If I don't know, I don't need to act. So, and with the IT, they helped me out with that. So hopefully, fingers crossed, it's going to work tomorrow. I try with some of my students, MSc students. Okay, could you do anything with it? I say, no, Mary, I can't even print that out. Nothing. I cannot do anything. So, but you can see it. Yes. So hopefully it will work. Okay. And that's the only thing that I can do. So I don't violate the copyright because that's what they do, right? They sell books. So, and I understand the situation that some of you are right now financially and all of that. If you were in person, I have some for those students that financially can't afford it. So I normally lend some of those books. And unfortunately, sometimes they don't come back. But in most of the cases, they come back, and I just try to share one or just keep it in the lab for the students to consult. But with this virtuality, that's the only thing that I can do. That's the best I can do. Okay, so you want to be able to open it, but no extraction, no printing, no saving, nothing. Okay, that's the compromise that we all need to do. So tomorrow we are going to do a couple of things, the pre-lab, and then we're gonna have, excuse me, that's not the order. The order is article discussion. For those that have taken Verfis with me, we always start with the article discussion. We'll see how it goes tomorrow. I don't know if I want to split you guys into rooms. I'm gonna make that decision tomorrow with the TAs. I love to be in the article discussion, but if I split the rooms, I won't be able to be in each room at the same time, the breakout rooms. So I'm gonna see how the dynamics. I'm very delighted to see that you are engaging with one another in perusal, because that was my objective. And I wanted just to ask you guys, how is your impression so far with perusal and this interaction in the groups that you have? Emma?
I actually really enjoy it. I think it's pretty interesting how we can see everybody else's comments beforehand, or sometimes like the questions that they ask, you don't necessarily think of them yourself. So it gets you thinking in a different way as well. So I really enjoy it.
Yeah, because all of you have different backgrounds, right? You have taken courses that differ from one another, and there are things that you maybe have seen in advanced chemistry courses that is, oh yeah, but nobody else has those things like that. Annika, because I see that you have been active, very active in there too. So
yeah, I find it's interesting to be able to like see what other people think about it. Just sometimes like if someone asks a question, we don't really know the answer, so we can't really help. But yeah, that's what I think.
But it plants the seed in there, right?
Yeah, for sure.
And there are some that has come and say, oh, yeah, I know, I have seen that. And I see that some have added some links to another paper. I read another paper, I searched. So it just to incite a little bit that critical inquiry, I think is important at this level at which you all are. Regurgitation has never been my friend in any of my courses. I hate regurgitation. I prefer to plant the seed in your brains of critical reading of things and relating things together. So by creating this group, that's one of the things that I want to achieve. One, that you interact with one another. Yes, Annika, yeah, sometimes you don't know the answer, and that's fine, right? But the questions are now there, right? So you're gonna be able to maybe ask, and believe me, I don't know the answers to all of the questions that you have, okay? I'm not the planarian expert here, and I will not pretend to be one. But it does allow to have this reading with a critical eye instead of reading just to obtain some sort of an answer to a question that I may ask, right? It's just to integrate the material. So we're talking about W and T now, the shift polarity, right? And Drake. And so it's like we're going to see these words again and again as we go through the course, and that's the main objective. I'm going to upload 2 other papers today or tomorrow for next week. And once again, remember, you don't need to read both. You just need to read one of them. And I hope with 60 students that we have people that are going to read either, right? And we're going to continue. The groups will be a little different this time, because every time that I assign, there's a different grouping that is made. Okay. So, and then once we do the article discussion, we're going to come back and do the pre-lab. Okay. So normally the article discussion, it depends on how much people participate into that activity, can be 20 to 30 minutes. And then we come back to our pre-lab, and that pre-lab normally doesn't last even half an hour. And after that you are on your own. And you have questions regarding anything, you can ask me in the lab session. And if that is all, 11:24, I'm gonna let you go. Please keep yourself safe. I will say go outside today. No, it's pretty cold outside, but keep yourself safe, and trying to have at least an hour of self-care is important. And Maher, you were working like crazy based on what you told me. So you gotta find at least 1 hour for yourself. It's important. You won't be as efficient if you don't. We always just go like that, and it would take an hour just to sip a coffee and just watch through the window or play with your cat or your dog or your snake or whatever pet you have, if you have. Try to do it. Self-care is important, particularly on the scenario that we are.
I totally understand, Professor, but I have been doing this for the past 16 years.
Maher, me too, and I learned how to find my time.
Yeah, I am trying to, but in order to graduate this course or this degree and then apply for medical school, I need to work very hard.
I know. I was there. I know exactly.
Because I was actually a gynecologist back home, and I worked 72 hours straight delivering babies and doing C-sections. But the problem is, here I have to start from all over again in order to...
I know. It's not fair. But
fine. I have to abide by the new country.
By the rules. Yeah, me too. I did it. So I know exactly what you're going through. But I tell you that you can learn how to find time for yourself. It's important. Half an hour.
Yeah. My wife is helping me do that. So I have.
Wives are good for some things sometimes.
Yeah, she's really a time management genius. So
okay, so good that you have a partner that supports you and forces you to take some break. It's important.
Yeah, she is very supportive. Yeah.
Perfect. Okay, guys, I'm gonna let you go because I just